We're going to start by looking at how prokaryotes regulate gene expression. So we're going to talk about some of the players in here. These things include activators, inducers, repressors. These are all going to be proteins that bind to the DNA. But to start with, let's talk about just regulation in general and some of the language we use. So when you talk about regulation of any type, there can be positive regulation, which is going to increase the rate, and negative regulation, which is going to decrease the rate. In the case of prokaryotes, we're going to be talking about the rate of transcription. Positive regulation in this context is going to increase the rate of transcription, and then negative regulation is going to decrease the rate of transcription. I might use some other terms here, like activation, that's another way of saying positive regulation, or repression, that's a way of saying decreasing the rate of transcription, negative regulation. So there will be proteins that have these names, activators, repressors, that are positive or negative regulators. Okay, so let's look at the structure of prokaryotic genes. Remember, we said that there are often operons with multiple genes in there that are all regulated by one promoter region, one regulatory region. So let's take a look here. In this case, there are 4 structural genes here, and these all get made in one big polycistronic mRNA, and they get translated into protein A, B, C, and D. So when we look at regulation, we're looking at control of transcription. So that's going to involve things that bind to the promoter, the regulatory region. You could have repressors that bind to this and literally block or lower the rate of transcription. Or you could have activators that come in and bind to this region and bring in RNA polymerase and increase the rate of transcription. We'll look at a couple specific examples of both positive and negative regulation here in this section. All right, so this is the basic structure. Let's look at a real example. So here's the Trp operon. This is a repressible operon. That means it can be turned off, basically. And the Trp operon encodes the genes needed to synthesize the amino acid tryptophan. So tryptophan is one of our amino acids, and there are 1, 2, 3, 4, 5 genes right here that are involved in producing tryptophan. We call these genes the structural genes. These are the genes that do the work in this thing. So this operon has a promoter, and then it has a region called an operator. I will talk about this in just a sec. Then we have the structural genes. So this is going to get transcribed all as one big long mRNA, and then the 5 different proteins get made. So the promoter, that's the region of DNA where the RNA polymerase is going to bind to. Next to the promoter is a sequence we call the operator. The operator is a region of DNA that can be bound by what we call a repressor molecule. When the repressor molecule binds to the operator, it sits there and it literally blocks the DNA, and RNA polymerase cannot move past that repressor molecule. We need that repressor molecule to be removed for RNA polymerase to be able to transcribe this. So let's look at the situations where it's bound or not bound there. Okay, so here is the pathway, right? We have our precursor that goes in, and enzyme 1 converts that into a second compound, and enzyme 2 converts that into another compound, and then enzyme 3 converts that into the amino acid tryptophan. Okay, so you'll notice this enzyme is coded by TrpE and D, this one by TrpC, and this one by B and A. So there are 5 proteins in here that make up 3 enzymes. Okay, so if we have a large amount of tryptophan, we don't really want to make tryptophan, right? We don't need to do that. So let's look at how tryptophan can be negatively regulating its own production. Okay, so here are the 2 possible states the cell could be in. And you might notice this looks kind of like logic and programming. It's very much a basic, simple kind of computer program, essentially. It's an algorithm. So we have 2 possible states. The cell has enough tryptophan to function, so it doesn't need to make more, right? If you have enough tryptophan, use the tryptophan you have, don't make more. All right, the other possible state is the cell is low on tryptophan, so it needs to make more. Okay, then we want to turn these genes on, right? So in this case, we don't want these genes on. In this case, we do want the genes on. How are we going to do that? We need some sort of switch that can sense these states. Do you have enough tryptophan or do you not have enough tryptophan? We need a sensor. Okay, that sensor is the repressor protein. So in the case, let's look where we have plenty of tryptophan. Here are my tryptophan molecules. When we have lots of tryptophan around, there is a protein called the repressor. The repressor binds tryptophan, and that allows it to come in and bind to the DNA. That repressor literally sits on the DNA there, and it's going to block RNA polymerase, preventing transcription. So no transcription occurs, none of these genes are made. That's okay, we have lots of tryptophan. So the repressor binds to the operator, blocks this, no tryptophan gets made, we're going to use what we have. All right, as that level starts to lower and lower, tryptophan goes away. The repressor protein, now that it doesn't have any tryptophan bound to it, falls off of the DNA, and RNA polymerase can transcribe these genes. As the level of tryptophan builds up, the repressor will come back and bind and shut this process off. So that's a really simple system, right? If we think about it, it's effectively the repressor protein that is sensing the state of the cell. It's sensing, is there tryptophan or is there no tryptophan, right? That's a sensor. That's a way of sensing the world. That doesn't seem super profound. It's like, yeah, it's on or off, but like, think about yourself, right? How do you sense the world and what does that do to your body? This is like the simplest example of that. So baby steps here, right? Okay, so here's the overview of the trip operon, right? When tryptophan is around, it binds to the repressor, which negatively regulates. It comes in and blocks the DNA, and the RNA polymerase can't transcribe. As tryptophan levels fall, RNA polymerase is allowed to transcribe these genes, and the proteins get made, and we can go on and make more tryptophan that the cell needs. So this is a negative regulation context, right? The trip repressor protein is a negative regulator. Okay, that's our simple example. Let's build a slightly more complicated example now. Okay, we have the sugar lactose. Lactose is the sugar that we find in milk. Lactose is a disaccharide made up of glucose and galactose. Okay, we're going to talk about the bacteria E. coli. Now, I know people think lactose and they think lactose intolerance. That has to do with humans and whether you make the enzyme to break down lactose or not. We're going to look at how E. coli breaks down lactose. So do not mistake what I'm going to talk about for something about lactose intolerance. If you want to learn more about gene regulation and lactose intolerance, take my Micro 250 class. We learn all about the enzymes involved in that and why some people are lactose intolerant and why some aren't. So, but we're going to focus on E. coli here. Okay. So, E. coli have the ability to break down the sugar lactose. To do this, they need genes, right? They need proteins. So, there is a operon called the Lac Operon that has 3 enzymes in it, lacZ, lacY, and lacA. This is what we call an inducible operon. So, instead of being repressible, it's inducible. We can turn it on. These 3 genes are needed to break down lactose. LacZ is the one that actually is going to break the bond between the 2 molecules here. So that's its function. Permease has a role in allowing lactose into the cell. LacA is called transacetylase. We still don't fully understand its role in the process. I've been teaching this for a long time. I've never quite found that we have a straight answer on exactly what it's doing in there, but it's part of the operon. Okay, so we need these 3 genes to break down lactose. So let's think about this from the opposite perspective. Let's think about it in the gene regulation perspective. When would a cell need to make these proteins? And when would a cell not need to make these proteins? It's actually a little more complicated than you think. So pause the video, think about these 2 contexts here. Okay, well, your first answer, you're probably thinking about lactose, right? Well, when would you need to make these proteins? Well, when lactose is around, you need to make the stuff to break down lactose. Okay, that's a good thought. When would you not need to make this? Well, when there's no lactose around, it's pretty useless to make these genes because they're not going to have anything to do. Okay, that's one side of it, but there's actually a second piece to this. Lactose is not the ideal sugar to use for metabolism. You remember back to the metabolism chapter, we talked a whole bunch about the sugar glucose, which is a monosaccharide. Well, these genes are actually going to break down lactose into glucose and galactose. So, if you have lactose present, it's all good and well to break it down, but what if you had glucose around? Wouldn't it just be better to use the glucose instead of making enzymes to break lactose into glucose, right? So, when there's no better sugar around, we also might need to make these. So this is an and situation. When lactose is present and there is no better sugar around, we need to make these proteins, right? We need to use the lactose. But when there is no lactose around or a better sugar, like glucose is present, we wouldn't need to make these. So even if there was lactose around, as long as there's glucose around, we don't need to make these, right? We'll just use the glucose. All right, so let's break this down into our sensor idea again. Well, what do we need to sense here? Well, we need to sense the presence of lactose, and we need to sense the presence of a better sugar, like glucose. So we're going to need two sensors here. So twice as complicated as the Operon, right? It's two things now. All right, let's look at the players here. Okay, here's our Operon, and here's our promoter and our operator. There's another gene that's involved in regulating this thing. It's called lacI. lacI codes for a gene that makes a repressor protein. So lacI gets transcribed into mRNA, which gets translated into the repressor protein. The repressor protein blocks transcription by binding to the operator. It looks the same as our case where we had tryptophan, right? Okay, so the repressor protein has a specific shape, and it actually has a binding site for the sugar lactose. When there's no lactose around, it goes into this shape and binds to the DNA in the operator region, blocking RNA polymerase. No genes are made to break down lactose. So, in our first scenario, right, no lactose around, there's no transcription. Okay, so let's look at what happens when lactose is present. Well, lactose comes in and binds to this repressor molecule. We would call lactose an inducer because it induces a shape change in this. We call this an allosteric change. Remember back to when we talked about enzymes, allosteric sites. It binds to the repressor protein and changes its shape. It no longer fits on the operator. That allows RNA polymerase to come in and transcribe the genes, and then they can get translated into proteins. So this is changing the shape of the repressor, right? So this is a little bit strange to think about, but removing a repressor is positive, right? It causes transcription to occur. Okay, so our lacI protein, the repressor protein, is our sensor for lactose. It's a negative regulator, okay? When no lactose is around, it shuts the process down. When lactose is present, it falls off and transcription can occur. All right, so that's sensor number one. But wait, we said, why break down lactose if there is a better sugar around, right? So why break down lactose into glucose if you already have glucose present? All right, so here we're going to look at the sensor for glucose, right? We need to know, is glucose present or is glucose not present? All right, how this happens is a little more complicated. Glucose levels are related to basically levels of ATP, and conversely, cyclic AMP. So remember, ATP is our energized state. When we cut two of the phosphates off, we de-energize it, and it forms a molecule called cyclic AMP. That's the low energy state. We would then need to burn energy to make ATP from this, okay? So Glucose levels are related to the level of cyclic AMP. When there's a lot of glucose around, we can burn that in our metabolism to make lots of ATP. That means there's very low cyclic AMP levels, right? If you have lots of ATP, the energized state, you have not a lot of the de-energized state. When glucose levels drop, you're going to start burning your ATP and you're not going to have anything to replenish it. So you're going to build up a lot more cyclic AMP. Okay, so why do I mention this? Well, there is a protein that's going to sense the level of cyclic AMP. This is called the catabolite activating protein, or the CAP. It's effectively a sensor for glucose, but it senses glucose through the level of cyclic AMP. Okay, pause your brain, think about that, right? Remember this. When glucose is high, there's low cyclic AMP. When glucose is low, there's high cyclic AMP, so they're inverse. Okay, let's fit this in here. So this is an activating protein. That means it's a positive regulator. When it's present, transcription will occur. When it's not present, transcription won't occur, or at least it won't happen very rapidly. Okay, so where does the catabolite activating protein bind? The CAP binds to the promoter region. Here's our operator sequence that we saw. lacI repressor bound there. The CAP binds here. Okay, CAP is inactive until it binds to cAMP. Once cyclic AMP binds to it, the CAP goes and binds to the DNA. And when it binds to the DNA, it brings in RNA polymerase. Okay, so it's a positive regulator. It's saying, hey, RNA polymerase over here, I need you to do transcription. So it recruits the RNA polymerase. So when there's high cyclic AMP, it's bound to the DNA. So when high cAMP levels are around, that means glucose is absent, right? When glucose levels rise, like here, when glucose is present, we have low cyclic AMP levels. That means that the CAP is inactive. CAP doesn't bind and it doesn't recruit RNA polymerase. So this is sensing glucose in this. Okay, so now we have two sensors. We have a sensor for lactose. We have a sensor for glucose. That's pretty clever, right? So let's look at what happens in the different states. Okay, so the Lac Operon has sensors for the lactose and glucose. lacI is the protein that senses the presence of lactose. The CAP protein senses the presence of glucose through cyclic AMP. If lactose is absent, lacI binds to the operator, repressing the situation, right? It blocks RNA polymerase. So the base state is if there's no lactose around, it is useless to make these genes. We're not going to do it. If lactose is present, lacI doesn't bind to the operator. That means transcription can occur. But there's a second question then, right? Do we need to make these? Because is there glucose or is there no glucose? So let's look at that second situation. Well, if glucose is absent, then yes, we do want to make the genes to break down lactose. So the CAP will bind and bring in RNA polymerase, increasing the rate of transcription. It's a positive regulator. If glucose is present, the CAP doesn't bind, and there's basically very low levels of RNA polymerase brought in. Now that's a little strange to think about because technically a little bit of transcription is occurring, but not a lot. So it's very, very low. We can effectively think of it as off. Here's a diagram that can kind of show you some of the different states here. This diagram is a little bit easier to look at. So let's look at this. Low glucose and lactose available. Well, CAP binds and our RNA polymerase comes in and there's nothing blocking it. So transcription is going to occur at a high level. Okay, let's look at this. We have high glucose, but no lactose. Well, that lacI repressor is going to bind and there's no CAP. So there's no RNA polymerase and it can't do transcription anyways, even if it was there. All right, let's look at a bad situation. Low glucose, low lactose availability. the genes are not going to be expressed. We don't have any lactose, so we can't afford to waste energy because we also don't have any glucose, right? So it takes energy to make these genes. We can't waste it, right? So everything is blocked. Everything is off. Here's the sticker, the kind of sticky situation. What if we have high glucose and lactose available? Well, there's no repressor protein bound, and there's not much cap bound, right? But there's going to be a super low level of transcription that will just occur. RNA polymerase happens to float by. It might transcribe it once, right? So it's not technically off, but it's not really on. And this is where our kind of thought of like a digital computer kind of breaks down, because it's not digital, it's analog. There could be no expression. There could be a little bit more, lots of expression, then back down, right? So it's a little more complicated than a computer program, right? Because we can have gradations. It's like a dimmer switch on a light. All right, so let's think about this situation. When you remove a repressor protein, what happens? I'm gonna pause the video, think about this. All right, this is a bit of a double negative. So repressors stop transcription. When you remove a repressor, that means transcription is going to start. I put this one in here because this is a bit of a mind-bending concept. I remember when I learned this was like thinking double negatives, right? Removing repressors starts transcription. All right, let's summarize what we've learned here. So we learned about the two types of regulation. Positive regulation increases the rate of transcription in prokaryotes. And we have activators. These are proteins that bind to the promoter. They recruit RNA polymerase to increase the rate of transcription. Negative regulation, on the other hand, decreases the rate of transcription, usually through repressor proteins that bind to an operator. They're going to block RNA polymerase from doing its job. We saw the Trip Operon. That was a repressible situation where we had a negative regulator. Tryptophan binds to the repressor protein, causing it to block RNA polymerase. When tryptophan levels drop, the repressor doesn't bind and transcription can occur. We can make tryptophan. Simple situation. We then looked at the Lac Operon, which encodes the genes to break down lactose. It has sensors for both glucose and lactose, right? Because we need to know the state of both those sugars. lacI was the protein that senses lactose. The cap protein, on the other hand, sensed glucose through CAMP levels. When lactose is absent, the lacI protein binds to the operator, repressing transcription. When lactose is present, lacI doesn't bind to the operator. It removes the repressor, right? Lactose bound to that repressor, changing its shape. Glucose, on the other hand, that is our positive regulator through the cap protein. Cap protein binds to the promoter and brings in RNA polymerase. When the glucose is present, though, the cap doesn't bind and no RNA polymerase is recruited. Okay, so those are our simple examples here. We're going to talk next about eukaryotes and how they have more places that regulation can occur.